Welcome to the Wednesday afternoon lecture. This, as it turns out, is the final Wednesday afternoon lecture of this academic year, as we are now going to take a bit of a summer break, but it's great to see all of you here for what I am quite sure is going to be a fascinating presentation from our presenter, Dr. Sandra Wolin. She is currently at the NCI, where she is chief of the RNA Biology Laboratory and senior investigator in the Center for Cancer Research, and also head of the Center for Cancer Research's RNA Biology Initiative. Her undergraduate degree was at Princeton. She went on to get an MD PhD at Yale, and we figured out that she and I were in the same building, the Sterling Hall of Medicine, at that time when I was a postdoc and she was an MD PhD student, doing her work with Joan Stites, the very well-known and very productive and wonderful professor of biophysics and biochemistry. Following a postdoc that she did with Mark Kirshner and Peter Walter at UCSF, she returned to Yale and there became assistant and then associate and then full professor in molecular biophysics and biochemistry, and also supported as a Howard Hughes Medical Institute investigator during that time. Then became the vice chair in the Department of Cell Biology and director of the Yale Center for RNA Science and Medicine. And just 2 years ago, we stole her away from the ivied halls of the Yale Medical School and brought her here to NIH, to the National Cancer Institute, where, as I've mentioned, she has a number of roles, but maybe particularly appropriately for today's presentation, chief of the RNA Biology Laboratory. She's an elected fellow of the American Academy of Microbiology and of the American Association of the Advancement of Science. And her research has focused in very interesting ways that you're going to hear about on a variety of interesting phenomena about cell biology, particularly the role of non-coding RNAs and how that might turn out to be an important clue to the pathogenesis of autoimmunity, particularly for lupus. And that is a tale which is still unraveling and revealing secrets in the research that she's doing, which I'm sure you're going to find quite interesting, involving such things as RO60 and commensal bacteria. So without further ado, please, would you welcome our WALS lecturer, Sandra Wolin.
Thank you, Francis, for that wonderful introduction. And thank you for inviting me to give this lecture. I've had a really fun day today, meeting people that I've wanted to meet for a while, even though I've been here 2 years. So one of my jobs here as head of what we now call the NCI RNA Initiative because we have expanded beyond CCR to include other branches of NCI, is to advocate for RNA and to help people understand how very important RNA is for biology and medicine. So I wanted to start by just talking about my favorite subject. So I took this from a review by Tom Check and Joan Stites called The Noncoding RNA Revolution. And 95% of RNA in cells is non-coding. And you know, if you took a biology course in high school, which you learned that DNA makes messenger RNA, which makes protein. And it turns out that RNA does a lot more than that. So now we know that there are all these non-coding RNAs in cells. This is an animal cell, and the RNAs don't function on their own. They assemble with proteins. And so we have small nuclear RNPs that form the spliceosome that splices pre-mRNA into messenger RNA. The telomerase RNA protein complex maintains the ends of telomeres. Link RNAs, long non-coding RNAs, modify chromatin and do many other things. And this is just, and in the cytoplasm as well, little RNAs called microRNAs can regulate translation and mRNA stability. And this is just the tip of the iceberg. There are many, many more non-coding RNAs and non-coding RNA protein complexes in cells. And most non-coding, and one thing about these non-coding RNA protein complexes is that most of the time the RNA has to be extensively folded and assemble precisely with proteins for the RNA protein complex to function. So, for example, this is the large subunit of the ribosome. It contains 2 non-coding RNAs. And this is the structure that was determined initially in Tom Steitz's lab at Yale. These blue squiggles are the non-coding RNA, and the green are the proteins. And you can see that it has to form this very complicated structure. This is a tRNA. It's also highly folded. And even little RNAs, like microRNAs, have to fold correctly in order to function. And so just as proteins misfold, RNAs misfold, and just like proteins fail to assemble correctly, so do RNA protein complexes. And so, just as they do for proteins, cells need a whole network of chaperone proteins to assist these processes, and also surveillance systems to find the RNA protein complexes, the RNAs that misfold or fail to assemble. And so this subject, how RNAs fold correctly with proteins and how cells find the bad ones and target them for degradation, has been the focus of my laboratory for a long time. And so what I'm going to talk about today is our work on this part of the pathway, RNA surveillance, how cells recognize the bad RNAs and target them for degradation. And what we know about this part of the process from many studies is that, the effector proteins at the end are ribonucleases, but that the ribonucleases are not sequence specific. So instead they rely upon adapter complexes whose role is to identify the misfolded RNAs and to recruit the nucleases. And so my laboratory began working in this problem basically in a roundabout way. What we set out to study was the molecular function of 2 autoantigens that were major targets of the immune response in patients with the disease systemic lupus erythematosus. And these are called the LA and the Rho60 autoantigen. And our method of approach was to identify these proteins in genetically tractable organisms and then use genetics to ask what they did. And so in work that I'm not going to tell you about today, we showed that the LA protein, which was known to bind newly synthesized RNAs, is actually an RNA chaperone that assists the folding of these non-coding RNAs and their processing and their assembly with proteins to form functional RNPs. And many of you know some of this story because Rich Mariah here is one of the leaders of this field. So what I want to tell you about today is our work on the Rho60 protein. And what I'm going to tell you is that it's actually an unusual adapter for recognizing defective RNAs. And it's unusual in part because it functions with a non-coding RNA called yRNAs. And so this is a study that really began with patient autoantibodies. So I'm going to tell you first about the disease, and then I'll tell you about our very basic work in animal cells and bacteria. And then at the end, I'm going to tell you about how our bacterial work has contributed to a new theory for how autoantibodies in diseases such as lupus might arise. So this story begins with patients. And so this is the part that starts at the bedside. And so the disease is systemic lupus erythematosus. And this occurs predominantly in women of reproductive age, and it can affect many different organ systems, which is why it's called systemic. The term lupus comes because the characteristic facial rash looked to early physicians like the bite of a wolf, hence the name lupus, and now we call this a butterfly rash. And so the insignias of many of the lupus organizations incorporate both the butterfly and the wolf. So one of the unifying laboratory findings in this disease are anti-nuclear antibodies. And so what these are antibodies that are directed against nucleic acid protein complexes, such as chromatin, the spliceosomal SNRPs, the Rho60 protein, which is also called SSA because it's also used for diagnosis of Sjogren's syndrome, and LA, which is also called SSB for the same reason. And so when I was a medical student and Joan Steitz's lab, in fact, was starting to unveil the molecular nature of these complexes, we were told time and again that these really had nothing to do with the disease, that these were an epiphenomenon, and lupus was really just a disease of the immune system, and the antibodies to RNA protein complexes, while tools for molecular biologists, were really of no clinical value, except in their role in diagnosis. And now we know that that's completely wrong. And the turning point was the discovery of endosomal toll-like receptors. So now we know that the antibodies actually play a central role in pathology. So somehow these intracellular autoantigens are released from cells, where they form immune complexes with patient autoantibodies. And these immune complexes can be endocytosed by cells that have a receptor for IgG on their surface, and then they land in the endosome, where the nucleic acid moiety activates an endosomal toll-like receptor. So toll-like receptor 7 binds single-stranded RNA, toll-like receptor 9 binds CpG-containing DNA, toll-like receptor 3 recognizes double-stranded RNA, and toll-like receptor 8 also sees single-stranded RNA. And so activation of the toll-like receptor results in a signaling cascade that results in increased type 1 interferon production and tissue damage. And this gives rise to the tissue damage, to some of the tissue damage in lupus. So most of the time, patients have antibodies against a lot of different autoantigens. And so it's not easy to tell if particular autoantigens are responsible for particular pathologies. But there are some symptoms that are invariably associated with anti-Rho60 antibodies, or anti-Rho antibodies. So anti-Rho antibodies, they're common in lupus and Sjogren's syndrome. And mothers with these antibodies have about a 2% chance of giving birth to a child with what's called the neonatal lupus syndrome. So the most serious manifestation of this disease is a permanent cardiac conduction defect. So the classic one is complete AV nodal block, and 20% of these children die in utero, and the ones that survive require permanent pacemakers, so cardiologists eventually see them, adult cardiologists. And these children also develop transient sun-sensitive skin lesions, and here's a baby. And so the skin lesions disappear when the antibodies can no longer be detected in the baby's circulation. So they've long been suspected to be pathogenic. And these antibodies are also strongly associated with a skin form of lupus called subacute cutaneous lupus, which is a highly photosensitive disease, and it occurs in many contexts. It can be idiopathic, which means we don't know why it occurred. It can be drug-induced, so the drugs that do it are some antifungals, antihypertensives, and even some cancer chemotherapeutics. And it also can be paraneoplastic, which means it occurs in the setting of cancer. And so anti-Rho antibodies recognize two structurally unrelated proteins, Rho60 and Rho52, But Rho52 is actually a ubiquitin ligase, and only Rho60 is a nucleic acid binding protein. So it's the only one that could be functioning in these toll-like receptor mechanisms. So the Bayan lab proposed that perhaps the reason that maternal anti-Rho60 antibodies are so tightly linked to congenital heart block is that they're actively involved. So in this scenario, so Jill Bayan is a rheumatologist who's head of rheumatology at NYU. What she proposed is that the antibodies get released somehow from dying fetal cardiac myocytes, where they can form immune complexes with the mother's anti-Rho60 antibodies that cross the placenta. And then they are taken up by macrophages, where the nucleic acid moiety activates toll-like receptor 7, leading to an inflammatory response and fibrosis of the AV node. And you can come up with a similar model for how Rho60 RNPs might be involved in a disease in subacute cutaneous lupus. So sunlight-damaged keratinocytes might stimulate the release of RNPs, which could bind patient antibodies and be taken up by plasmacytoid dendritic cells and activate toll-like receptors. So another reason that anti-Rho60 antibodies are of clinical interest is that they're actually the earliest detected autoantibodies in patients who eventually go on to develop clinical symptoms. So I told you that a defining feature of lupus is the presence of autoantibodies against nucleic acid protein complexes. And it turns out that some of these antibodies can be detected in patients many years before diagnosis. In fact, 1/3 of the women who give birth to babies with the neonatal lupus syndrome didn't even know they had autoantibodies, that when the congenital heart block is diagnosed in utero by ultrasound, then they're tested and found to have them. And so it turns out that on average, the earliest detected antibodies in patients who will go on to develop clinical symptoms are against Rho60. And the initial epitope was defined, and this was done in John Harley and Judith James's labs. And what they proposed was that these antibodies originate by cross-reaction with an Epstein-Barr virus protein called EBNA1, because there's a lot of epidemiological data linking lupus and Epstein-Barr virus infection. But there's no primary sequence or structural homology between this primordial epitope and EBNA1, and I'll come back to this point later. And finally, as I try to convince you that Rho60 is worth studying, we collaborated with Richard Flavel's lab some years ago and made a mouse lacking Rho60. And remarkably, the mouse developed a lupus-like syndrome, complete with autoantibodies against chromatin and ribosomes. And it had the characteristic glomerulonephritis. This is a section through a normal mouse kidney and through a knockout mouse. And this pink stuff is the deposition of immune complexes in the glomerulus. And this is the same. And these are the EMs. EMs are the same. This is looking at the EM level. Here are the deposits of IgG. This is the same lesion that lupus patients get. So what this means is that the cellular function of Rho60 may be important for preventing against autoimmunity. So what is Rho60? So this is a crystal structure that we did with our collaborator Karin Reinisch at Yale. It's a ring-shaped protein. It's a monomeric ring. And it's present in most animal cells. Remarkably, 5% of sequenced bacterial genomes and some archaea. And its best-known RNA partners are non-coding RNAs called yRNAs. So humans have 4 yRNAs. They're called H for human, Y, and then named, and there is no Y2, Y1, Y3, Y4, Y5. And so they all form these lollipop structures and with a long stem. And within the long stem is the Rho60 binding site, and they all conserve the 5' portion of the stem. And the protein, we actually have co-crystals with this portion of the stem and row. And so this portion of the stem is in the crystal structure. The yRNA binding site binds on the edge of the row 60 ring. And invariant residues in row 60 that are conserved all the way to bacteria make specific contacts with invariant nucleotides on the 5' part of this strand, the ones in red. So it's a highly, highly conserved sequence-specific RNA binding site. But what you'll see here is that in this space-filling model, you can see our beautiful hole. It can accommodate single-stranded RNA. And so the question was, what binds in the hole? Well, over the years, before we had a crystal structure, my lab had some kind of strange data that row 60 might actually be involved in binding misfolded RNA. And we discovered this first studying Xenopus laevis row 60 RNPs. Basically, my very first postdoc, Charles O'Brien, who's now a professor at the University of Arkansas for Medical Sciences, decided to cut apart a frog and immunoprecipitate from each of the different tissues. And what he found was that in Xenopus ovary extracts, there was an extra band in our immunoprecipitate. And it turned out to be a large collection of variant 5S ribosomal RNA precursors. We went to the literature. We discovered that Xenopus had 20,000 oocyte-specific 5S genes, and there was no selection against mutation with so many genes. Many of these things were variants, and our protein was binding to a collection of variant RNAs that misfolded. And then later on, we showed that row 60 bound variant U2 small nuclear RNAs, which is an RNA in the spliceosome, in mouse embryonic stem cells that also seem to be misfolded. And now, of course, with modern techniques, we have greatly expanded the known targets. So what do I mean by misfolded? This is the universal 5S ribosomal RNA structure. All 5S RNAs from bacteria to humans can fold into it. The nucleotide changes in our variants were disrupting conserved helices and allowing the formation of alternative helices. And so Gabi Fuchs, who is now an assistant professor at the University at Albany, took this on as her thesis project. And she showed that this portion of the pre-5S was necessary and sufficient for binding, but that the sequence, it was not sequence specific. She could flip the helix, it bound just as well. All it needed was helices and a tail. Our friends in the Reinisch lab got a co-crystal with an RNA consisting of a helix and a tail, and found out that the tail went through the hole and out the other side. And the helix lies on the row 60 surface, and all the interactions are with the RNA backbone. So, which explains the lack of sequence specificity. So what Gabi showed was that pretty much any RNA with helices and a tail would bind row. So what we have proposed is that row 60 may function as a scavenger, that rather than recognize a specific feature of a misfolded RNA analogous to hydrophobic patches on the surface of misfolded proteins, that row 60 binds any RNA with a protein-free single-stranded end and an adjacent protein-free helix. And these RNAs are protein-free because they have failed to assemble with their correct RNA binding proteins because they're misfolded. So that took us some distance, but there were many, many questions that we needed to know that were still unanswered. And one of the big ones was, what do yRNAs do? And do they play a role in this process? So one thing we know is that yRNAs can function as tethers. So row 60 binds to this portion of the stem, And the other portion of the RNA binds to what we call effector proteins, proteins that do something. Having different tops of the RNA allows a different complement of proteins to associate. So Yan Leng in my lab has made mouse cells that are deleted for either, using CRISPR deletions, for either the mouse Y1 RNA or the mouse Y3 RNA. There's only two mouse RNAs in two mouse Y RNAs, or cells lacking both. And so this protein ZBP1 only binds if Y3 is present. It's not present when Y3 is knocked out in the co-IP. And this RNA helicase, MOV10, requires the Y1 RNA for binding. So they form specialized RNPs based on their effector proteins. We don't yet know the role of the MOV10 helicase in Rho60 function, but we do know that ZBP1 is an adapter for Rho Y3 RNA nuclear export. So Rho and Y RNA shuttle in and out of the nucleus. If you deplete ZBP1, Rho accumulates in the nucleus, and so does the Y3 RNA, as shown by fluorescent in situ hybridization. So we know that one role of that Y-RNA-mediated tethering of Rho60 to ZBP1 is required for nuclear export. But what we really wanted to know was how Y-RNAs contribute to the role of Rho60 in non-coding RNA surveillance. And our biochemistry suggested that the rest of the RNA lies across this surface and could not access the whole because there's no free single-stranded end. This end is down here. So what we thought is that Y-RNAs might be regulatory, that they might function somehow to gate access of the whole to other RNAs, but we had absolutely no idea how this could be regulated. So we decided that we really needed a single-cell genetically tractable organism to learn more quickly, to be able to combine biochemistry and genetics, and that's why we study bacteria. So my goal when I started my lab was to find Rho60 in yeast. When the Cerevisiae genome came out, we stopped looking. Then the Pombi genome came out, and it wasn't there either. And at that point, the first bacterial genome started to come out, and lo and behold, there was one with a Rho60 protein. So we now know that it's more than that. So many bacterial genomes contain a likely Rho60 ortholog. And so these include most bacterial phyla and both pathogens and commensals. And so, but at the time, we didn't know this. All we knew was that there was a single bacterial sequence with a Rho60 ortholog and that it might be our only chance to work on Rho60 in a semi-genetically tractable system. So we went forward. And the bacterium was Deinococcus radiodurans. It's the world's most radiation-resistant organism, which is why it was an early sequence. And Xingwo Chen, who's now a staff scientist in my lab, took this on. And he showed that Deinococcus lacking Rho60 were less resistant to UV. We went back and looked at our mouse embryonic stem cells lacking Rho60, and they were less resistant to UV, so it seemed to be a good model. We showed that Rho60 and Y RNAs are upregulated after a bunch of stresses. And we went back to our mouse cells, and they weren't upregulated, but they moved from the cytoplasm to the nucleus after UV or oxidative stress. And then Xingwo showed that Rho60 functions with a ring-shaped exoribonuclease called polynucleotide phosphorylase to degrade ribosomal subunits during stress, and that there was a complex that he could purify containing Rho60, PNPase, and yRNA. And so, to make a long story short, he reconstituted this complex from purified components. He showed that this is the Deinococcus yRNA. There's actually two RNAs in Deinococcus. This is the one that's part of the complex. Rho60 binds to this conserved region, and that this binds to single-stranded RNA binding domains at the top of this ring-shaped nuclease. And so we collaborated with David Taylor and Hongwei Wang, who were at Yale and now at Tsinghua, and Hongwei moved to Tsinghua University to do single particle EM on the reconstituted complex. And what we found is that we had a new double-ringed RNA degradation machine that was scaffolded by non-coding RNA. So this is the Rho60 ring. It's tethered by yRNA to this ring-shaped exonuclease. It's configured such that single-stranded RNA can pass from the Rho ring into the nuclease cavity for degradation. And Tsinghua went on to show that this complex, which we called RIPER for Rho60 yRNA, PNPase, exoribonuclease, RNP, was specialized to degrade structured RNA. It was better than PNPase alone with a structured RNA, less of the input transcript, more limit oligos in this degradation assay, and this is just a graph of the data. So to summarize what we learned from our Deinococcus studies, we learned that tethering of Rho60 to a ring-shaped nuclease specialized it for degradation, and that's because normally the nuclease uses these single-stranded RNA binding domains to bind single-stranded RNA. And what I forgot to tell you is not only is this complex better at structured RNA, it's worse at single-stranded RNA than PNPase alone, and that's because the binding surface of PNPase is now replaced with a binding surface of Rho60 because a yRNA is interacting here. And so this surface is specialized to bind single-stranded RNA, and the Rho60 surface is specialized to bind RNAs with helices and tails. So, yeah, and the other thing was we learned things about what yRNAs do. So they can function as both tethers and gates. So in the absence of an effector protein, they can function as a gate to keep other RNAs from the whole. And we had genetic evidence in bacteria that was one role. And then they could also function as tethers to tether Rho60 to an effector protein, in this case a nuclease, to create a new RNA degradation machine. So by now we knew that there were many other bacteria that had Rho that were far more genetically amenable, and we have moved our studies to Salmonella typhimurium, which is very similar to E. coli, and you can do practically anything with Salmonella that you do with E. coli. And so in Salmonella, it's encoded in what's been called an RNA repair operon. This is the E. coli version. It's called an RNA repair operon because it has this RTCB RNA ligase, whose function in our cells is to ligate tRNA halves after intron excision. So some tRNAs are made with specialized tRNA introns, and there's a specialized tRNA splicing machinery that removes them, and this is the ligase that ligates them back together. And bacterial tRNAs don't have introns, so it's still an open question what the role of this is. So we identified yRNAs here, and they turned out to be encoded right next door. And we called them YRL for yRNA-like because they seem to represent a different family of yRNAs. They weren't quite as metazoan-looking as the Deinococcus RNAs that we had characterized. And so one thing that fell out of this was we ended up finding a lot more yRNAs. So it's very easy to find the Rho60 protein in a genome. Anybody can find it. You translate the protein. The homology is obvious. It's very hard. It can be very hard to find a non-coding RNA in a genome because what can be conserved is structure, not sequence. So the only yRNAs that were known were the ones that we had laboriously characterized by making antibodies to the protein, immunoprecipitating, looking at what was there, sequencing. And so for a long time, we had tried to use bioinformatics to go further. So this is a Deinococcus yRNA. And if you blast this, all you get are other dynococcal Y RNAs. But it turns out if you do a low stringency blast on the Salmonella one, you start seeing other sequences, and they're always encoded either 5' or 3' to the Rho60 protein. And you can fold them into similar structures. We built homology models, so we could search for the secondary structure using a program called Infernal. And when we were finished, we had identified Y RNAs in more than 250 bacteria and 20-something phages. And in every case, they were always encoded either 5' or 3' to the Rho60 protein and transcribed in the same direction. And then we could look at what was common. And we found that we could draw these with these conserved stem loops. And so I've labeled them D and T and AS. And we could find this so-called T loop in the Dinococcus one, but not the others. And the reason I've given them these names is that the harder we looked at them, the more they look like a tRNA. So tRNAs fold into this cloverleaf structure. This is the T-arm. It's named because of the modification of this U, U is modified to a thymidine. The D-loop is named because it contains dihydrouridine, and this is the anticodon loop in the acceptor stem. So they look like circularly permuted tRNAs with a closed acceptor stem. So, and in this model, all 7 of these tertiary interactions can form. So we did what we thought was a good test for a molecular biologist / geneticist, which is we checked whether some of these nucleotides had the characteristic tRNA modifications, and they did. So this is dihydrouridine, and this U here, this is the T1, and this U is pseudouridine. And they were put in by the very same enzymes that modify tRNAs. So for me, that was a satisfying answer that, yes, this was true. But every time I presented this, a structural biologist would want to know if we did the structure. So finally, we collaborated with Yang Zhang, and we did a structure. And lo and behold, it is in fact true. So tRNAs fold into this L shape, and here's our tRNA domain, tRNA-like domain. And this tRNA, this L shape, is held in place by the very same tertiary interactions as tRNAs. So this is called the elbow, and it's formed by the interaction of the T loop and the D loop. This base pair is conserved in all tRNAs, and it brings together the D loop and the D arm and the variable loop, D loop and variable loop, and this is a characteristic base triple. So seeing is believing. So just to summarize what I've told you so far, so our studies of Rho60 and Y RNAs in animal cells revealed that it's a ring that binds misfolded RNAs in its cavity. And we learned that Y RNAs can function as tethers and gates. So Y RNA tethers a bacterial Rho60 to a ring-shaped nuclease, forming a double-ranged RNP machine. And so a big question now is how this relates to the role of Rho60 in mammalian cells. And now most of my lab is, again, studying Rho60 in mammals. By performing homology searches using the Salmonella one, we have found that this particular RNA, YRLA, is widespread, and that it's a new class of non-coding RNA that mimics tRNA. So now we're not just talking about, we have to say tethers, gates, and tRNAs, but we don't yet know what the role of the tRNA-like domain is. So now I want to switch topics and go back from the bench and this very basic work to lupus. So one of the questions that was raised by our identification and characterization of Rho60 in bacteria was whether commensal orthologs of the human Rho60 protein might actually be important for triggering autoantibody formation in susceptible individuals. And this was a collaboration with Martin Kriegel a rheumatologist and immunologist at Yale. So I told you that a defining feature of lupus is the presence of antibodies against nucleic acid protein complexes. Some antibodies can be detected actually up to 10 years before diagnosis. And the earliest detected antibodies on average are against Ro60. And this is the initial epitope. And so in 2005, when this paper came out, I read it, and there was the epitope they were pointing to in EBNA1 that was supposed to be important for cross-reaction looked absolutely nothing like the Ro60 epitope. And by then, we knew that there were bacterial Ro60 RNPs. And so it seemed possible to me that instead of invoking an unrelated protein, that maybe the bacterial Ro60 proteins had something to do with it. And so from 2005 to, I think, 2013, every time I talked to a rheumatologist, I told them my theory. And I have to say that nobody wanted to hear about it because this was before the microbiome was hot, and people have their own things they're studying. And one day I had the luck to go to a junior faculty retreat at Yale as a senior faculty mentor, and the person sitting next to me said his name was Martin Kriegel, and he was interested in the role of the microbiome in triggering autoimmunity through molecular mimicry. And I said, oh, I have a great story I want to tell you. And I did, and he immediately thought it was interesting too, and so we started working together. And so this is that B-cell epitope that was defined in this paper, and these are 3 commensal bacteria. So P. propionic and C. amiculatum are in the skin, and Aspergillus maciliensis is in the gut. And you can see that that epitope is hugely conserved. And this is a T-cell epitope that was shown in mice in Shuman Fu's lab at University of Virginia to be important for epitope spreading, which is a phenomenon where you initially make antibodies to one part of an RNA protein complex, and they spread to other determinants on the protein or the macromolecular complex. So, and this just shows you where this epitope maps on our structure, and it does indeed overlap the RNA binding domains. So commensal bacteria containing Ro60 are all over our bodies. And so these are the commensal niches, and these are some of the commensals that contain Ro60. So the first thing that Martin's lab asked was whether specific groups of bacteria whether the microbiomes of patients with anti-Ro60 were different in some way from normal patients and patients that don't have Ro60. So this is just 16S sequencing, healthy patients, lupus patients without Ro60 antibodies, and lupus patients with them. And they looked at fecal or mouth and skin microbiomes, and basically everybody was the same. But this, within each phyla, there's only a few members that contain Ro60. And so what they did next was a candidate organism approach. So they looked at the levels of 4 different common Ro60 commensals and found that most of them were the same too, but that there were 3 patients that had elevated levels of the skin commensal P. propionicum in their chest skin, and this guy that's much higher than everybody else, this swab actually came from one of a lupus skin lesion. So, and they could demonstrate that patients with subacute cutaneous lupus erythematosus, which is invariably anti-Ro60, they looked at 7 patients, and 5 of them had P. propionicum in their lesions as demonstrated by in situ hybridization. And they were not found in psoriasis lesions. So they also asked whether patient T-cell clones could react with both antigens. So these is from a T-cell clone library made from a Rho60 positive patient plasma. And so they selected T-cells, T-cells that are stimulated by P. propionicum bacteria, and they looked at CCR6- and CCR6+ T-cells because CCR6 is thought to be a marker for T-cells involved in autoimmunity. So they took the ones that were stimulated by P. propionicum and found that a number of them actually are also stimulated. These clones are actually stimulated by human Rho60 protein or by the T-cell peptide that is implicated in epitope spreading. And then my lab took these antibodies and we tested whether we could immunoprecipitate bacterial Rho60 RNPs using them. So this is P. propionicum. It's not a genetically tractable bacterium. We didn't have antibodies to monitor the immunoprecipitations, but we had bioinformatically identified their Y-RNA. So what we did was we immunoprecipitated, and then we isolated RNA from the immunoprecipitate and ran a northern blot and probed for the P. propionicum Y-RNA, which is here along with some precursors. And so using this assay, we were able to demonstrate, ultimately using about 30 different anti-Rho60 positive sera, that about 80% of them cross-react with the P. propionicum RNPs. So next we wanted to take this beyond correlation and try to get at causality. And so we teamed up with Andy Goodman at Yale and made gonadobiotic mice, so mice that were monocolonized for a specific Rho60 commensal, Bacteroides theta iodomicron, which is a common gut commensal that contains Rho60. And this was done in 2 strains of mice. And what was found was that the mice that were monocolonized with B theta developed antibodies that detect the human Rho60 protein on ELISA. And as a negative control, mice were colonized with Enterococcus gallinareum, which doesn't contain Rho60, and they did not develop antibodies. So this is at least some evidence that commensal Rho60 proteins could and can trigger autoreactivity. So just to summarize this part of the talk, I told you that commensal bacteria containing Rho60 orthologs are widespread, and that some Rho60 positive patients have increased levels of P. propionicum in their skin lesions, and some patient T-cell clones react with both the P. propionicum and human Rho60, and we can immunoprecipitate Rho60 RNPs from bacteria using patient antisera, and that mice monocolonized with a Rho60 positive gut commensal produce antibodies that cross-react with human Rho60. So we're proposing a new mechanism for the etiology of lupus autoimmunity, which is that in genetically susceptible individuals, commensal Rho60 positive bacteria could initiate and drive lupus autoimmunity. And so one of the questions this raises is how widespread is this? Does it apply to diseases outside lupus? And I have to say we don't know, but I thought that, well, since this is the home audience, so to speak, I should speculate wildly. So a lot of these, there are a number of systemic autoimmune rheumatic diseases that are associated with antibodies against distinct nucleic acid protein complexes. And I've told you about Sjogren's and subacute cutaneous lupus. And if you blast the human protein sequence against a database. These are the closest commensal bacteria. Now, some patients with myositis make antibodies against the signal recognition particle, which is an RNA protein complex I didn't have on my first slide. It targets nascent secretory proteins to the endoplasmic reticulum. And these antibodies are highly associated with pathology. And so what's interesting to me is that signal recognition particle in humans has 6 proteins and an RNA. The bacterial one has a single protein, and that is the one targeted by the antibodies, which is exactly what you would expect if it had a bacterial etiology. And you can go on with other autoantibodies. So Jo1 is against the histidyl tRNA synthetase. It's true that everybody has one. We have to charge our tRNAs with histidine. But some commensals are closer to the human protein than others, and so on. So this is something I would like to pursue here with collaborators, which is why I thought I would discuss it. But just to close, I just want to thank the wonderful collaborators that I've had. So Adam Stein and Karin Reinisch did our initial crystal structures of the Ro60 protein. David Taylor and Hongwei Wang did the single particle EM on the Ro60 PNPase complex. Wei Wang and Yang Zhang did the crystal structure of the tRNA. And our work on lupus autoimmunity was carried out with Martin Kriegel's lab. And so a lot of what I've showed you was done in my lab at Yale over the years, but I've now moved to Frederick. And so our work has always been funded by NIH, both outside and inside the walls. And our study on the role of the commensal bacteria in autoimmunity was funded by the Arthritis Foundation. So thank you.
Well, thanks, and appreciate the provocative ending there that puts forward other hypotheses. It's time for questions. People, please, if you have questions, come to the microphone so the people in the video audience can hear as well. And let's begin right over here.
Thank you very much. Very nice talk. And good for you. You can do it in the lab. My intellectually maybe driven question is, the ring composition, I don't know what it is, but are they charge differences in the bacteria or in the human, and whether they are affected by some kind of metals, whether there is charging within that holds them the way they do, from within or from without, and anything infective agents or aluminum in the vaccines or so forth can damage them that leads to autoimmunity. It's a very detailed topic.
So I know of no evidence linking vaccines to autoimmunity, but metals are of course important in RNA protein structures. Magnesium, for example, stabilizes RNA interactions.
Great, thank you.
Over here.
Well, since you found some bacterial orthologs, so you think fecal transplantation in the mouse model could help to reduce this autoantigen and autoantibodies?
I'm sorry, what is the question?
Fecal transplant, since you found some orthologs, do you think getting bugs from healthy individuals or mice could take care of the autoimmune disease in mice or later in human?
So I think you're asking about the possibility of using this therapeutically. And so I think that what we would need to know is exactly which bacteria really are most tightly associated. So, our approach was a candidate bacterium approach. I think we really need to look at the microbiomes of patients using full sequencing of genomes so we know exactly which bacteria we might be talking about. And if we could do that, I think it would be possible to design therapeutics.
All right, thank you. Good luck.
Over here on the right.
Yeah, thank you very much for your talk. I think you very convincingly show that these antibodies can be raised towards these commensal proteins. However, I'm curious, do you still believe there's a requirement for the Rho and perhaps LA as well to get on the outside of the human proteins, to still get it on the outside of the cells to be recognized for the toll-like receptors? And then, why are those proteins out there? Well, what's the role for biologically? Why would they be just thrown about?
The antibodies are on the outside, and the antigens are intracellular antigens. And there's a lot of reports in the medical literature that when cells become apoptotic, the antigens can access the surface through a pathway that really hasn't been described, okay? And so I was always a little afraid to touch it, but we collaborated with Jill Byon's lab a few years ago to try and ask this question. And what we found was that we did this in mouse cells where there are only 2 yRNAs. And what we showed was that the Y3 RNA was required for row 60 to reach the surface of the cell and become accessible to antibodies during apoptosis. Okay, and we could show that the Y1 RNA would not substitute. We could rescue it. We did everything we could think of, and that was the result. So I believe there's some protein that binds the Y3 loop and takes it to the surface through a pathway that has not really been described yet.
So a question from the point of view of the bacteria, I guess. So do you think in the rest of the bacteria where you don't see this kind of function, there's something parallel that's doing the same kind of quality control? And will it have an RNA tether, or will it just be straight?
Well, our favorite candidate for redundant function in yeast, for example, and also bacteria, would be the ring-shaped complexes of SM-like proteins. And of course, the bacterial one is HFQ. And so we've always wanted to do genetic experiments to ask that question. We just haven't done it yet. Rich.
Nice talk. So as you know, LA antigen hasn't been found in bacteria, but in yeast. And so do you think there are yeast commensals that might account for it?
So I think what the rheumatologists and immunologists would say is that the anti-LA antibodies develop later. They're usually linked to anti-Rho, so probably the precipitating event is the development of antibodies against Rho 60 and then epitope spreading to LA.
One more.
My question is about the knockout of the Rho 60 in the mouse. There's no antigen, so why is there lupus there, do you think?
Very good question. So I forgot to tell you that one reason, so we don't, the short answer is we don't know, okay? But I can speculate. So I forgot to tell you in the beginning that one reason that RNA surveillance pathways are important is that it's possible that they protect cells from being activated. Intracellular RNA sensors like RIG-I and MDA5, that they prevent them from being activated by excess endogenous RNA. And so we got this result in 2003 when the only known RNA sensors were the toll-like receptors. And so we were trying to say, gee, maybe excess intracellular RNA matters at a time when we weren't ready to think about it because the other piece of the puzzle, right, the cytosolic sensors had not been described. But I think that's a question that should be asked.
Very good. So let us again express our thanks while heading to the medical library. Those of you interested in coffee and cookies, you can continue the conversation, but please let us thank again Professor Wolin.